the Institute for the Study of Peak States presents the second International Psycho-Immunology and Psychobiology Research Symposium. Developing a Psycho-Immunology Treatment for Covid-19, presented by Dr. Grant McFetridge.
Dr. Grant McFetridge, I'm sure just about everyone who's listening, certainly live, is the founder of the Institute for the Study of Peak States, has a electrical engineering degree from Stanford, PhD in psychology, and we wouldn't be here if it wasn't for him. One of the things that you may not know about him is how altruistic he is. So I was actually with Grant about a year ago when we were starting to see the early signs of COVID, and he was like, we have to do something about this. We were only just starting to get psycho-immunology understanding, and he could see that it was possible that we might be able to come up with a psycho-immunology treatment for that. And he just wanted to do it because he could see that there was possible that millions of people were going to die for this, and he wanted to be able to do anything he could to change this. So I'm going to hand you over to Grant, and he's going to tell you about the journey for over the past year. Thanks, Grant.
Thank you, Shane. Can you see and hear me?
Yep.
Excellent. Okay, let me just get this going. No, just a moment. Somehow we've messed up. One more time. Let me 1 second. There we go. Did that work? Can you see my screen?
Yes, it can. And so hit play and it'll go, yeah, that's it.
Got it there. Great, thanks. All right, so this talk is on our work on developing a psycho-immunology treatment for COVID-19. And let's make sure that it works. There we go. Okay, so I think we should just start the talk with an update on how it's actually working right now, our treatment. And it's looking good for people with active COVID or a long-tail COVID. It seems to get rid of the symptoms really fast, usually within an hour to, depending on the length of the treatment, up to 4 hours. So it's pretty amazing. I mean, people can be really sick, and then we do this little process, and boom, all their symptoms go away. What's interesting also is that some people who have been sick, their symptoms temporarily come back during the treatment and then disappear again. So it sort of suggests that people are still infected, and it's just gone into a kind of a background state. However, we've been working with clients who require oxygen, and they're a different set. Their lungs are really in bad shape, and until recently, we had made absolutely no success, made no difference what we did. And just recently, we think we've cracked that problem, and we're working on to simplify that thing that we solved. And I'll talk about that in the talk. Okay, so we have a web page, and you can keep updated on our progress at peakstate.com/covid19.html. And if you have COVID, especially if you are requiring oxygen, please give us a call at grant@peakstates.com, because we need people to test the newer treatment on. Okay, and I'm going to start out by acknowledging the team. When the COVID hit, we just stopped everything and started working on this project. And I sucked in every person in the Institute that had any ability to work on this. I want to thank everyone. We put thousands of hours into this project. We weekends, evenings, I mean, the whole, people actually have jobs and such. And so they sacrificed a lot of months of their own lives, many months, to work on this. Well, I also want to thank all of the COVID volunteers, the people who contacted us with COVID and were willing to act as experimental subjects. Okay, so I'll quickly mention safety and liability concerns. What I'm talking about here is not something you should be testing on yourself. This is for educational purposes only, and there's distinct safety issues that we as researchers deal with that you shouldn't be trying to do on your own. You simply don't have the background or the understanding that's needed for this sort of stuff. If you want to acquire some of that background, you can read some of the textbooks we've written, particularly subcellular psychobiology. So what do I mean by safety? Well, our work in psycho-immunology is very much like working in a drug company. You are in the research phase and you experiment with making a drug. And maybe it works, maybe it doesn't.
Shane, does it change slides for you guys?
No.
No, it's not actually.
Uh-oh.
So there's something here.
Something's broken. Because it shows that it's changing for me. Huh. Oh, technology. I'm going to stop sharing. It says paused. How do I make it go, do you think? One moment, folks. Resume share. You like that? No, that didn't make any difference. It says it's paused, but it doesn't say why. Shane, are you doing something at your end?
No.
It's my screen is locked up.
Maybe
allow me to do
stop
anything.
sharing and then try again.
Okay. Do I come back? Nope. Don't even come back.
Well, now you're back. And
Okay. Can you guys see anything?
Yeah, we see you.
Yeah.
Okay, good. How about desktop too? I want this one.
well, choose that one and try again. And then play.
Can you see that, guys?
Yep.
Yes.
Now, did that show up?
it has.
What's your screen say?
Yeah, let me tell you this detective story.
Okay, so let's come back to here. Can you see this one?
No, it's not changing.
It's not changing? Huh. Okay, well.
Oh, now it, no.
Stop sharing. I have no idea why this is happening. Does this work? Can you see something now?
Mm-hmm.
Here's the title.
Mm-hmm.
Okay. So I am going to just do this.
Yeah, and just go through them individually. Yeah, that's a good idea.
Does that work? First slide, second slide, did it change?
Yes.
It's working.
Okay, good. Well, here's the people that I was acknowledging, thinking that you guys were seeing this. Safety and liability, this is for educational purposes only. So don't experiment on your own. And safety testing, this is where I was starting to start. We spend a lot of time safety testing. And that's because at the research phase, it's very much like doing drug research. You don't know if it works. You don't know what the dosage is. You don't know if it's going to even help. So that's the phase that I'll be describing here. And then there's the testing phase. And that's where we test on healthy people to make sure that there's no weird things that happen that we weren't expecting. And then finally, we cautiously release the therapists. And then from there, we release the project fully. Okay, so at this phase, we're at the research and testing phases. And we've been iterating through the two, as you'll see shortly. Okay, so now that we actually have a working screen, I'm going to tell this like a detective story because I'd like you to follow along and think about what you would have done in our circumstances. But fortunately, we've taken out the thousands of hours of effort that it took to generate these little slides. Okay, so how would you investigate coronavirus? Okay, first of all, we don't have any money. We do this volunteer and we don't have access to the very expensive lab equipment that so many other researchers in the world have. So, but fortunately, we have a history of working on diseases, and we were able to apply this to COVID. So years ago, we figured out how to see inside both people's bodies and see inside the cells. And as I write here, I used to think this was all bullshit too when I heard people could do this, but we've figured out how to do this reliably, and we use that for our research. Mostly it's cost. It's cheap and easy to do this. Okay. The problem with seeing inside your cells is it doesn't come with little subtitles. So you actually have to have a biological knowledge and background to understand what you're seeing, and which is one of the reasons why when we first discovered that we could do this sort of thing, we didn't realize we were seeing inside cells. We just didn't have any references from our own work background. Okay, but the bottom line is, if this stuff is real, then it should be able to generate treatments that actually work. And I think that's true in everything in life. I mean, you take your phone. Does this actually work? Well, it's using electromagnetic waves that nobody can see, but it works. So they must be there. So it's the same with our work. Okay, so we started on March 4th, and we got really lucky right off the bat because one of our research staff had actually gotten COVID. He was right in the beginning of the plague, and it was great because he knew our tools. So we used him as an experimental subject. And by March 11th, we hit our first real big surprise. We saw that the coronavirus that infected him was actually carried by 2 different amoeba. And it's interesting. There should be one on the hand there, and there's one through the air.
Down, see if it comes in.
Ah, thanks. So, so we'd never even heard of this, but yet we could perceive that they stuck to his hand and his arms, came into his bloodstream by the hand, and we could see that it came in through the nose, or he could in this case, and into his lungs. And we actually went scurrying back to the literature to find out that yes, this does happen. Although interestingly enough, as far as I know, at this point, nobody in the COVID research community is talking about any of this. So you and we have to take this with a grain of salt. Is this real? Well, we think it is, but we just went from there and said, okay, if it's real, we need to generate treatments that take this into account. Okay. So on March 21st, the first breakthrough. Now, you should understand that we really had no clue how to do psycho-immunology well at this point, or even at all, except for one successful treatment on a fungus years ago. So our first breakthrough with COVID was finding out that these amoeba were emitting a toxic fluid into his body, and that it was affecting him. And so this, on March 21st, we found the first one, and it was coming from the airborne amoeba. Okay, so we went, if that's the case, then what do we do with this? How do we get rid of this? And the next major breakthrough in psycho-immunology occurred here when we realized that that toxic fluid being given off by the amoeba was similar to a fluid that we're supposed to have very early in development. So let's just, again, I'll say that here's the present with the toxic fluid, here's the past with the fluid you're supposed to have. And because of trauma, it turns out, the body says, oh, I need this. And mistakenly wants the fluid that the parasite, the pathogen, is giving off. Okay, so that was a fundamental breakthrough. This is fluid microevents. This whole concept of fluid microevents happened on March 28th. Okay, on March 30th, we found something even weirder. And to this day, I'm still puzzled about this. When we were looking at this fellow's body and going back in time with regression, we found that there's what felt or he experienced as little worms inside his body from his belly to his eyes. And when he saw a person who had COVID, these worms got really active. And then we found that someone who was sick with this COVID disease, you feel sick, you're tired, and you don't want anyone to work with you. Well, it seemed to be a function of these worms. They were somehow influencing the psyche to cause people to leave them alone. One of our therapists who had an active case was saying, "Oh, I'm just sick. I don't want to do any research. Leave me alone," like that. And it turned out it seemed to be related to this weird body worm thing. Where this is in the primary cell, I don't know. But anyway, next one. On April 13th, we found something else weird. We found that the coronavirus itself emitted fluid out its spikes, and we never even heard this is possible. To this day, I don't know if it's anywhere in the literature, but that's what we were seeing. So something like that. Okay. On April 15th to May 2nd, once we had these micro events, we spent time just really healing them to find out what the heck was supposed to happen there. Why did they go wrong? What was causing us to want these fake fluids later on in life? What was the reason that they could even go wrong? We're doing this for safety because we need to understand everything that we're doing around this process. And also, if we could understand what happened and what went wrong, maybe we could figure out simple ways to heal it. After that, on May 13th to 17th, we started our first round of safety testing. And to do that, we used a bunch of our therapists, actually the worldwide community of certified therapists, and asked them if they'd volunteer to test this process. What we found is that our success rate varied between 65 and 84, depending on what kind of fluid we were working with. If you look at this little table, the mean treatment time was about an hour to an hour and a half, but it just wasn't good enough. Now, we were measuring this because we had found something. Let's see if I go back here, somewhere around here. We had found that if you heal this very early developmental event, people start to experience a kind of a sparkling in their body, a fizzy sparkle, almost like if you drink fizzy water on a sunny day. So we had at least a measure for monitoring, are they actually succeeding in healing this stuff? And we weren't doing as well as we would have liked. Still, if you're sick and this works, that was okay, but I'd like to be seeing stuff in the 80, 90 kind of % range. It turned out that all this testing, and we spent a lot of time on this, turned out to be wasted effort, and you'll see that shortly. Okay, on May 25th was a bummer of a day because we realized this idea that getting rid of toxic fluids wasn't getting rid of the pathogens. Essentially, all this effort that we put in safety testing and all the rest was wasted effort. On May 28th, we settled down after being so disappointed and went back into the lab and made a new breakthrough. And we realized that we had successfully gotten rid of the fluids from the infection. And that actually allowed us to perceive the membranes of the organisms that we were working with. And that was a huge breakthrough. And that led to a successful PI, psycho-immunology process. Okay, so we're back on track. And we could then take that membrane sensation and track it back to a fluid micro event in development. Okay, so now we're all excited and we do some more safety testing. And at this point, things are working better. We're seeing a 82% success rate using the sparkles, internal sparkles criteria. Okay. So June 12th, we had focused on the virus on that last one that I just showed you a slide for. We'd gotten the viral membrane and we got rid of the viral membrane, but we realized we had mistakenly thought it got rid of everything, but we finally realized that it didn't get rid of the amoeba, but it did work on the virus. So it wasn't a complete waste, but we needed to do something better because the amoeba themselves were giving symptoms. Okay, at that point, from June 15th to July 29th, Dr. Lykkegaard and myself had to leave home. And we were out of touch for basically 6 weeks. We didn't get back till around August 1st. This was not a good time because we were right in the thick of the COVID testing, and we had patients, and I mean clients, volunteers, I should say. But just life is the way it is. Okay, so August 1st, we get back to work. And on August 7th, we get 2 new COVID clients, both who are using oxygen. So we go, okay, we've got this treatment, let's give it a try. We watch as the virus seems to be getting rid of, and 2 things happen. One, it doesn't seem to affect their symptoms. They still seem to be drowning. They feel bad, and the other thing is, it looked like healing the virus was releasing something from the interior of the coronavirus. Let's see. Okay, so by August 14th, we've tried our best. We're a complete failure on the oxygen. We think we've gotten rid of the virus, but we can't fix the need for supplemental oxygen. So what the heck is happening? Let's see. Good. One moment. Okay. So we dig back into these clients, and they're great troopers. They're feeling not well, but they're willing to work with us, and for that I'm just incredibly grateful. So when we actually do the psychobiology of the disease, we find something really strange, and that is the people who are infected, seriously infected, they feel like they've got this kind of hungry monster. I believe it was off to the right side. Yeah, off to the left side. And it's kind of floating in space over there, and it's very strange. And then we also discover, as we're trying to figure out why these people are suffocating, that to them, it feels like their chest is filled with water, like they're a barrel with water inside of it. And that leads us to understanding more of the biology than we'd ever imagined was going on. Okay, so more weirdness. What we tracked that sensation of the monster to was a bacteria stuck on the side of the primary cell that came in from the amoeba that was carrying the COVID virus. And it would give a psychobiology experience to the patient that there was something threatening on their side. And the lung organelles also appeared to be involved somehow in this. We didn't even know there were lung organelles until that point, but there was a correspondence between how sick they were in their lungs and how these lung organelles were appearing. The lung organelles appeared to be attacked or damaged in some dynamic between this monster and the feeling of suffocation. Okay. So you can see at this point, we're just going, what the heck is going on? Why is this so complicated? Okay. So on the August 14th through 19th period, we go, let's try something different. Instead of just tracking the membrane quality, let's look at the entire picture. So what does that picture have? Well, we've got this bacteria, we've got these amoeba, and we have about 5 minutes left. I'll continue. So we throw out the simple membrane idea and we go into matching the symptoms. And sure enough, we find something. Back in development, just before conception, we find a moment that feels to the client the same as what they're experiencing with the COVID infection. And it's a very complicated moment in development, and things are really going wrong here. It seems just before conception, everybody and his brother figured out that they can infect the new zygote. It's like a window of opportunity for attacking the egg. So if you look at this picture, you can see that there's sperm almost to the egg. There's an egg, there's a polar body forming, and there's these funguses that are getting into the egg at this point. One we call the stabbing fungus because it actually acted to stab into the egg and damage it. One that looked like a vortex that was spreading itself in via the solar plexus. And there was, so we were healing and working at this point, and we realized that the virus, the coronavirus, feels like sperm to the person, like a whole bunch of little male presences. And so each of the things that we've been experiencing in the clients turned out to have an analog in this particular moment. The bacteria that stuck to the side, it actually felt like the polar body to the person. So you can see when I say psychobiology, there's a real link between the perceptual, psychological, and physical perceptions and the biology that are going on in this event. Okay. On September 23rd, we see that there's sexual feelings showing up in this developmental event that seems somehow related to COVID. And in fact, when we looked at people who got infected and they regressed to their infections, if they were around a super spreader, that super spreader felt very sexual to them. And so it seemed like the virus somehow interacting with the super spreaders. Maybe these people normally had a really high sexual vibe, but whatever reason, sex seemed to be involved in this problem. Okay, so November 2nd to 5th, we're still stumped. We've got this developmental event. It's very complicated. Uh-oh, I got 5 minutes left. Thanks. And now we're seeing viral variants. With people who volunteer to work with us, the quality of their membranes are starting to change from the original one that we started considerable time ago. Okay, January 2nd, another staff person gets COVID. We try healing sexual fluids that we're finding in this developmental event, and their symptoms go away. So what's going on here? January 14th, we're working on suffocation client again. We eliminate a bunch of these funguses and things that we're seeing back there. And as we clean up this event in ourselves, because it turns out that this event only sets people up for getting COVID, most people have these problems. When we clean it up in ourselves, we see stuck on the side of the egg, there's this little purse-like thing, and it turns out to be an amoeba with viruses in it. That's kind of the first breakthrough we've had, the latest breakthrough, because at that point we realized that the whole COVID infection, the amoeba, the viruses that were coming from the external environment in the last couple of months to a person, were reflecting this event that happened just pre-conception. Okay. So going back to this picture, you can see on the side there where I say the amoeba with the virus is stuck to the egg. Okay, February 7th, we get another breakthrough. And this is really important because this event that we're looking at, trying to fix the COVID with, is so complicated. There's so much going on. It'd be really impossible to have an easy fix at this point. So, but we figure out that if we focus just on the amoeba, that we could find a different developmental event to get rid of that. And that, getting rid of that little amoeba stuck on the side with the viruses in it, seemed to echo right up to susceptibility into the present. And we could do that at a different, more simple developmental event. Okay, so I'll just repeat that. The little amoebal sac that's on the side of the egg in development is a great marker for COVID immunity. Okay, so we're all excited and we've got our new treatment, and it fails again on some new clients who need oxygen. But yet it works great on everybody else. So something is different with the oxygen clients, which is baffling. On March 3rd, one of our staff gets the Pfizer vaccine, and it was really great because she knew these tools. She could watch the progress of the vaccine itself. What happened in her is that the vaccine causes her cells to express the same quality as the membrane in the COVID, and it's out of her body in less than 15 minutes. I think it was around, I can't remember if it's 5 minutes, but I said 15 to give it an upper range. But anyway, whatever we'd been doing with our work just cleaned that vaccine right out of her system. Later, we had one of our volunteers get the Pfizer vaccine and had a completely different experience. Without having our treatment, it lingered in his body for as long as we were watching it, and slowly expressed over time. So our treatment was doing something pretty different as far as the vaccines go. Okay, on March 10th, Dr. Likegaard comes up with an easier way to heal the trauma around the amoeba event. And on 14th, we start another round of safety testing. But we're still wondering, how the heck does this work as far as the suffocations? Like the suffocation clients, they have a different disease. It's like normal people have COVID, and then these people seem to have something else that's been turned on by COVID. And we get lucky. We find that we're finally able to see that the alveoli in the lungs are being damaged, and they're being damaged by fungus. And that seems to be what's causing the suffocation problem. And so we optimistically called it the suffocation fungus, because we've thought we've solved this before. We'll see how it goes. But fortunately, and if you look at our little picture, it turned out there was a suffocation fungus entering the egg at the same event. It turned out we hadn't noticed it until just then. These things are hard to spot, especially if you don't know they're there. Okay, let me go back to here. God, I guess I'm out of time. Yeah, that's where we are today. We think we've found what's causing people to suffocate. We tried it in a couple of people. It wasn't a perfect treatment. They still had some of the fungus left, but the suffocation symptoms really dropped. So we think we're on the right track. Now we just have to tweak the technique to include the suffocation fungus. Yeah, and that's where we are today. So again, here's the picture of what's happening in this development. We're not using this developmental moment anymore, but it's very handy because we can keep an eye on the amoeba with the virus to see if people are immune. Just the last little plug, we've written books on our work. I know this stuff that we're talking about sounds completely unbelievable. It certainly did to me, and I'm the one who developed a lot of this. But over the years, as we've worked, we've kind of kept a log of how we're doing this and how it's possible, and our models and our successes and our failures and such. Let's see. Okay, I guess there's no time for questions, but you can keep an eye on COVID process on our website, and you can also email me at grant@peakstates.com especially if you've got COVID, and especially if you're on oxygen. We'd love to make sure that we've really solved this problem. Long term, we hope that we just can put a very simple treatment up on the web so people can just use it. We'll see how that goes, but that's my hope for this particular disease. With the vaccine coming out, the need for this is fading. I just wish we could have solved psycho-immunology faster. It took us a year, and that's just the way it is. If we'd known what we knew now a year ago, we could have whipped it out pretty fast. Okay, I think that's it at this point. Shane, would you like to come on back?
Yeah, so there isn't any questions, just notes of gratitude, and that it's absolutely amazing the work that we're doing. So, but there might be some questions come through that you can respond to in form later. So thank you very much, Grant.
Thank you for watching. For more information, visit peakstates.com/covid19.html.